Hi, Professor Gronlund here. Let's talk about bacterial endospores. If you think that animal hibernation is a cool trick, you just wait until you hear about bacterial sporulation. An endospore is a completely dormant structure. Some bacteria can make them. Endospores are like a state of suspended animation. They don't carry out any life processes, no metabolism, no reproduction. Endospores are extremely resistant to environmental stresses, such as UV light, radiation, desiccation, boiling, freezing, chemicals, etc. Antibiotics have no effect on them. And it appears that time also has little effect on them. Bacterial endospores from the abdominal content of an extinct bee that was preserved in amber for 25 million years has been revived, cultured, and identified in the lab. Now, how is that for cool? The existence of these heat-resistant structures was first hypothesized in the mid-1800s by Ferdinand Kahn when he saw Bacillus subtilis growing on cheese after he had boiled the cheese. The ability to make endospores seems to be limited to several genera of gram-positive bacteria. Endospore formation, called sporulation, is usually triggered by the lack of nutrients. To start the process, the bacterium's DNA is replicated, and then the cell's plasma membrane pinches off between the replicated chromosomes, forming a spore. In the core, DNA tightly coils on small acid-soluble proteins that provide UV protection, and diplycholinic acid, a spore-specific chemical, dehydrates the spore to maintain the dormancy. Several protective layers then form, including a cortex containing modified peptidoglycan that provides heat resistance, and a spore coat made of proteins that provide resistance to many different chemicals. The endospore is released once the vegetative cell that generates it dies and disintegrates. The process takes several hours, but the result is a stable, dormant structure containing the potential to regenerate into a fully functional cell. In the same way that sporulation is triggered by the absence of certain essential nutrients, germination of that endospore occurs when those nutrients are available again. Basically, the endospore rehydrates, loses its protective coats, and recovers its typical shape and structures and functions of that living cell. Under optimal conditions, germination can be completed in just a few minutes. So, if you're now thinking, well, who the heck cares? I don't plan to revive 10,000-year-old endospores from a bee's gut. Well, perhaps not. But if you're going into a career in healthcare, you will still need to understand the significance of endospores. For example, you may have patients with life-threatening foodborne illnesses from eating leftover rice or improperly canned vegetables. Clostridioides botulinum endospores can survive improper canning, and Bacillus cereus endospores on raw rice survive the regular cooking process and will germinate while the leftovers are unrefrigerated. Both of those endospore-forming bacteria secrete powerful exotoxins. And then there's Clostridioides difficile, or C. diff for short. Sure, the antibiotic treatment takes care of the live cells initially causing the symptoms, but antibiotics don't hurt its endospores, so they germinate after the treatment is over and they cause the disease to recur. So, yep, if you're going into healthcare, you should know about endospores. In the microbiology lab, when we simple stain bacteria in that Clostridioides or Bacillus genus, we often see small, uncolored oval shapes inside those rod-shaped cells. Those ovals are endospores, and it should not surprise you to find out that in addition to all their other resistance, endospores are also resistant to staining. But in the 1930s, two microbiologists in Middlebury College in Vermont, Alice B. Schaefer and McDonald Fulton designed a procedure that's now frequently used to stain endospores. The procedure uses malachite green stain with steam for 10 minutes to stain those endospores and then a counter stain of safranin to stain the vegetative cells. In our lab, we place a staining tray with water in it on a heating block to create that required steam. You will need to wear your gloves and safety goggles while you're steaming with that malachite green. As with most staining procedures, you will start by using aseptic technique to make a bacterial smear. While the smears are air drying, turn on the heater so that the water in the tray is steaming when the smears are ready. Once the smears are dried and heat fixed and the water in the slide tray is steaming, put on your goggles, place the slides on the rack, and then use forceps to cover the bacteria with a square of filter paper. Now add the malachite green. Stain for 8 to 10 minutes. During that staining, whenever that filter paper starts to look dry, add more malachite green and add more water as needed to the tray. After 10 minutes, turn off the heater and use a clothespin to carefully remove the slides to a new staining tray. Use forceps to remove the filter paper and then thoroughly rinse that slide. Now counter stain with that safranin for 1 to 2 minutes. Thoroughly rinse and then blot dry. As always, remove the live bacteria from your workspace, disinfect your workspace, remove your gloves, and wash your hands before getting out your microscope to view your new slides. The heater with the slide tray can be left on the bench for now while it cools down. If things have gone well, your image on oil immersion should now show consistently sized green endospores and larger pink vegetative cells. After looking at your slides and putting your microscope away, carefully use the back of your hand with a quick light touch to see if the heater and slide tray have cooled enough to finish cleaning up your workspace. So now what do you think of endospores? Pretty neat survival trick, huh? And when the going gets tough, those that can make endospores. Work safe and happy staining.